Thank you, Sean, and welcome everybody. I hope that we have a productive hour in front of us looking at the use of soils information to talk about a variety of things, including species deployment decisions and how we might make fertilizer prescriptions and perhaps other questions that you might have. Now, if we talk about, and really the focus of my discussion today is principally in the southern pine region, so throughout the southeastern U.S. And if we look at this region in general, we can see really across the landscape there's an array of management practices from extensive to intensive. The extensive would be found more typically on public domain lands, Forest Service, state lands, and so forth, whereas the intensive lands may be on non-industrial private landowners, but more commonly on industrial kinds of ownerships. If we look at the trend in what's happened relative to southern pine plantations over time, it's a pretty impressive figure. You can see that we had less than 2 million acres of southern pine plantations in the 1950s. And if we progress through time to nearly present day, we have almost 40 million acres of southern pine plantations in the U.S. South, which is a remarkable feat over a relatively short period of time. As you might expect then, the U.S. South is certainly an important breadbasket for the United States and the world. I've got a basic question here I want to ask you to begin with, and the question is this: is site quality a fixed attribute for a given soil type? And if you'd use your polling bar and answer A, B, or C, that would kind of put us into a common realm. Okay, looks like some responses are coming in. So we have kind of a mixture. We have some that think that it is a fixed attribute, others not, and some that can't recall. That's all fine, and I hope that the next hour we'll be able to clarify whether or not this is a true statement or not. From my perspective, site quality is not a fixed attribute for a given soil type. As you'll see, we can have some pretty marked differences in terms of what can happen to a site relative to productivity through some of the silvicultural interventions that we use. So, next question for you. Is the potential productivity of loblolly and slash pine being realized in our U.S. South? Again, poll A, yes, B, no. Simple question. And as we wait for those responses to come in, most of you saying that the productive potential of slash pine is being realized. And I hope to impress upon you in the next hour that we're a far cry from really realizing the potential productivity of loblolly and slash pine in our region. So here's an example. This is not the U.S. South, but one that we certainly strive for. This is loblolly pine growing in Argentina, and the person in this slide is in excess of 6 feet tall, so you get some appreciation of what a 1-year-old tree would look like. By 2-year-old, there's a lot of leaf area in these stands. They're certainly occupying the site quite quickly. And by age 4, that site had already been pruned and thinned. Question is, are we approaching this level of productivity in the U.S. South? Quite honestly, we're getting closer, but we're still not at this level. This slide does a nice job, I think, of kind of showing you what's happened through time with regard to technological advances in southern pine silviculture. What we're plotting is volume at harvest on the y-axis in terms of cubic feet per acre, and we have time on the x-axis, and on the second y-axis we have the rotation age of that stand. And if you look at this particular graph, you can see the yellow bars represent what a natural stand would grow through time. So there's a fixed amount of growth potential associated with natural stands. By adding different technological advances like tree planting, you can see in the blue line that we've increased productivity simply from that activity by controlling spacing and stocking. The red in this case is additional advances in growth that we would expect from site preparation. In this case, we're talking about primarily mechanical site preparation. So bedding would be an example of a mechanical kind of site preparation. Fertilization has also played a very significant role in the U.S. South since the 1970s to current day. And you can see that it's generally causing an increase in volume growth over time. Also contributing to increases in productivity really since the 1970s is some form of weed control, and we're talking about primarily chemical weed control, either pre-plant chemical site preparation in more recent times or herbaceous kinds of weed control at earlier times. Tree improvement is also, typified by the purple bar, is also a very important component of the success story in the U.S. South relative to producing more productive, disease-free kinds of plantation systems. And then finally, in the last decade or two, there's been a lot of interest in biotech kinds of advances and varietal forestry, and that also has the potential to increase our harvest yields. So if we look from the 1940s to kind of current day, there's been more than four-fold increases in total volume production. Also shown in this slide is the reduction in rotation length associated with these technological advances. So in former times, 1940s, with principally natural stands, we would have been approximating 50 years for a typical southern pine rotation. Today, we're really looking in the upper teens and low 20s for a lot of our rotation ages. So this is a major success story in the U.S. South, and it again kind of reinforces the notion that you can work with the same soils, but through these types of silvicultural interventions, be it from genetics or chemical site preparation or fertilization, we can have pretty profound effects in terms of growth potential. So to kind of put us into perspective, really one of the main factors that limits southern pine productivity in the U.S. South is soil nutrient supply. And we've had a lot of work done since the really 1960s and 70s to current day looking at soil nutrient limitations. And largely what we've seen in a lot of our sites is it's competition for below ground site resources, either in nutrients, water, or both, that have a profound effect in terms of the potential productivity at a given locale. We can ameliorate some of these nutrient limitations with some of the silvicultural treatments that we use, such as site preparation, fertilizer additions, and again, understory competition control. All of those work part and parcel in terms of alleviating some of these nutrient stresses that the trees experience. Today, a lot of our nutrient management, and really in contrast to the 1980s, our nutrient management today is now practiced on a rotation-long basis. So we think about that and integrate that into our decision matrix really from day one. You heard in our previous webinars the importance of soil mapping. Ed O'Brien gave a very nice talk yesterday on land classification and soil mapping, and Daniel Markowitz kind of got us grounded in the first webinar talking about soil properties. But really, if we're trying to get into making site-specific silvicultural prescriptions, which is a goal for I would say virtually all of us, we have to do it using some type of a basis, and soil mapping certainly puts us into that realm of possibility. It's still not perfect, but we can get a lot closer than if we use some general blanket prescription. The CRIFF soil groups is one example of kind of putting soils into a management context that was introduced yesterday, but let me reintroduce it today in case we have new viewers. The Cooperative Research and Forest Fertilization Program at the University of Florida established these CRIFF soil groups in the 1980s. And the premise was to try to put soils into kind of an understandable, interpretable form for land managers. If somebody said, well, I'm looking at an Altic aliquot, a lot of us will roll our eyes and say, well, I think I've heard that term, but I don't really know what it means. If you're a soil scientist, you know it would be a soil that belongs in this CRIFF C group soil. So let me just kind of walk you through these very briefly. What distinguishes these soil groups is certainly drainage class. So we go from very poorly into somewhat poorly drained soils for the A's and B's, all the way to excessively drained for the G group soils. These would be the dry sandhill kinds of sites. Also, the H group soils are very poorly drained, and those tend to be the organic soils. They have a histic or organic epipedon, and a little bit more difficult for us to work with in terms of intensive forestry. But really, the A's, the B's, the C's, the D's, the E's and F's would fall into the realm of soils that certainly meet some of our goals relative to intensive management. The A and B group soils are primarily differentiated by where the clay subsoil or argillic horizon comes into play. If it's 20 inches or less and it's very poorly drained, a gumbo kind of soil, it's going to be an A group soil. If you're also in this very poorly to somewhat poorly drained realm and the argillic horizon is deeper than 20 inches, we would classify it as a B group soil. The C's and the D's are quite different taxonomically. These are Spodosols, and you have of course, a sandy surface horizon. You have a well-developed spodic horizon in both the C's and the D's. And what differentiates these two soils is the presence of an argillic horizon. So if you have any type of clay within 60 inches or so of that surface, it would be a spodosol with clay. That's a C group soil. If you bore down through the soil and you don't find an argillic horizon, but you do find a spodic horizon, it would be typically classified as a D. The E's and the F's are kind of the upland corollary of the A's and the B's, except they're moderately well to well drained. Oftentimes they're going to fall into the ultisol kind of category of soil orders, but the E group soils have clay within 20 inches of the surface. So if you auger down and you find that, chances are if it's well drained or moderately well drained, it's going to be an E group soil. F group soils would be clay deeper than that. And as we said earlier, the G group soils tend to be the sandhills with very little horizonation, probably going to fall into the entisol soil order. There's some generalities that we can make regarding important properties like soil drainage class and site index, in this case an example for loblolly and slash pine. Now, if this is a conceptual kind of graph, there isn't a numeric value here on site index, but in general, we find that there is a pretty good relationship between site quality and drainage class, going from excessively drained, well-drained, moderately well-drained, somewhat poorly, poorly, and very poorly drained. So our southern pines, loblolly and slash pine, seem to do quite well from a site index, an inherent site index perspective at somewhat poorly to poorly drained end of the moisture spectrum. We can also use indicator plants, and now that question came up in a previous webinar, and can we use indicator plants to kind of help us appreciate drainage classes? And the answer is absolutely yes. Of course, some of it is going to be a little bit dependent upon your topographic region. But in this case, if we're working in flatwood sites, typical of the CRIFF C and D group soils that might have spodosols, we tend to find that Gallberry, as an example, Ilex glabra, is a pretty good indicator in general of drainage classes. You don't really find any Gallberry in the excessively or well-drained soils, and it's really not until you get into the somewhat poorly and poorly drained realm of drainage that you end up seeing a fairly significant amount of Gallberry in the understory. So again, we use these kinds of tools as foresters and land managers to help us appreciate kind of the landscape position. And as you heard earlier, also in the earlier webinar, that as little as a foot of elevation difference in these types of soils can make and have a pretty profound effect in terms of site index. So here's an example in this slide that kind of shows you that spectrum. We're going from moderately well drained here, where there's a little bit of palmetto and a little bit of Gallberry here, to a better representation of both species, in this case in a somewhat poorly drained, and certainly well represented in the somewhat poorly, well represented in the poorly drained. The other thing that I like to look at to help appreciate drainage class differences from moderately well to poorly or even very poorly drained is the height of the understory plants. Fedderbush and Gallberry are going to be considerably taller in the poorly drained end of the spectrum than in the somewhat poorly drained. So again, little tools that you can use to kind of appreciate some of those subtle site differences as you work in the field. This diagram, we won't have a quiz on this at the end, but it is a useful key to help you appreciate how we might deploy different species relative to, in this case, the different CRIFF soil groups. So if we start off with the CRIFF A group soils, each of these gumbo clay kinds of soils, poorly drained, very poorly drained soils with clay very close to the surface. For all intents and purposes, these are going to be really best relegated to loblolly pine if that's within your management objective. By contrast, if we look at the C's and the D's, and these are the Spodosols either with clay or without clay, you can see that depending upon the drainage class and the amount of rust hazard, we could conceivably go from slash pine, if you don't have a very high or low to moderate rust hazard on a well to moderately well-drained soil, slash pine might be the species of choice. Or if you have a high incidence of fusiform rust, and there are maps that show incident levels across the South, then slash or longleaf might be your option. Going to the other end of the spectrum, over here at the CRIFF G group soils as an example, these are those excessively drained deep sands, and depending upon the site index, it might help trigger where you might consider species options. So if you have less than site index 40 and you're in these excessively drained soils and you're within the species range, sand pine might be a possibility. And if the site quality or site index is a little bit better, then longleaf pine might be a possibility. So it's a tool that you can use. You can modify it to your own conditions, but it generally puts you in the ballpark to assist you with choices for species deployment. So coming back to our question about factors that limit southern pine growth, I think these next series of slides are certainly going to focus on nutritional aspects. This isn't a poll, but the question is which nutrients typically limit southern pine growth? And I list nitrogen, phosphorus, potassium, manganese, copper, and boron. Quite honestly, all of these could limit southern pine growth on some sites. Nitrogen and phosphorus tend to be, of course, our most significant in limiting macronutrients. And some of the micronutrients that we have interest in and evidence of deficiencies are manganese and copper, and there's some evidence that boron might also be a consideration, but certainly manganese and copper. So I present this just as a refresher for most of you. When we start talking about essential nutrients required by plants to complete their life cycle, we break them up into two categories. Macronutrients would be those that are required in relatively high amounts by the plant, in excess of 1,000 parts per million, and these are the familiar ones that we all probably learned in basic biology: NPK, calcium, magnesium, and sulfur. By contrast, micronutrients are required in relatively small amounts by pines, and some of the micronutrients that certainly are essential are iron, manganese, boron, copper, zinc, molybdenum, and chlorine. We're beginning to see a little bit more evidence of micronutrient deficiencies once we've satisfied macronutrient demands in some of our stands, especially with multiple rotations. We'll come back to that in a later slide. This slide, I think, does a nice job to let you appreciate that concept that we talked about earlier, that nutrition management is a rotation-long kind of concern. If we think about current annual increment on our y-axis and age, we can see that there are distinct stages of stand development that might trigger the need for nutrient ameliorations. As a stand is developing, particularly young stands, a lot of energy of that tree is put into crown development, and also there's a significant amount of understory competition that's taking place in these young stands. So nutrient demand tends to be relatively high. As the stand closes canopy and some of the mineralization of the organic matter left after harvest, mineralization process slows down a little bit in terms of converting organic nitrogen and phosphorus to inorganic forms for uptake, we see that soil supply kind of declines. And other processes like internal resorption and recycling kind of increase in significance relative to meeting the nutrient demands of that stand. So we could also expect that we might see nutrient deficiencies or opportunities for fertilizer response in this stage of stand development, as in stage 1. Additionally, as that stand gets older, there is additional immobilization of nutrients that occur in the litter and plant biomass. And once that nutrient is tied up in that plant litter, it's not going to be breaking down and really converting into inorganic forms for uptake very easily, because the moisture and lighting regimes have changed, the microbial community might have changed as well. So there certainly could be opportunities for nutritional responses in category 3 as well. I like to use the barrel analogy to kind of let you appreciate limiting factors, and I'm just using a variety of soil factors here to kind of depict that. And some people might refer to it as Liebig's law of the minimum. But generally, productivity is going to proceed and move upward until some factor becomes limiting. And that would be the lowest hole in the barrel stave. In this case, it might be nitrogen. Well, what can we do? We can fertilize that site, and that might temporarily plug that hole in the nitrogen limitation and ultimately allow that stand to grow to its next level of limitation. And it might be another soil factor, maybe restricted rooting depth, or it could be phosphorus supply, or both. The second cartoon on this slide kind of shows you in a hypothetical way soil phosphorus supply and stand development over time. So relative amounts of nutrient supply relative to stand age. And you can see early in a young stand, the supply of phosphorus in this case tends to be relatively high, because again, after harvest you have organic debris left on site, you have some incorporation of that debris into the mineral soil, and mineralization processes take place. Some people might refer to this as an Assart effect. So initially there's fairly good supply of nutrients. But as we said, as these stands close and soil supply rates diminish a little bit, it's going to decline with time. At the same time, we know as trees get bigger, their nutrient demands increase. And there becomes a point in time where the demand, in this case, exceeds soil supply. So we might expect, in this case, to have a phosphorus deficiency present in this portion of the curve. And that would trigger our interest in making some type of ameliorative treatment, in this case, perhaps with phosphorus or nitrogen and phosphorus both. So these are kind of basic concepts that we use to help think about soils and growth potential and what we can do as land managers. There's a variety of diagnostic tools that we can use to kind of help us key into kind of managing the nutrition of a site. Again, either through fertilizer additions, weed control, or combinations of both. We've already mentioned the kinds of soil groupings, and the CRIFF groups are just one example. Drainage class is very important in terms of appreciating potential responsiveness and potential productivity. There are soil maps available, either private maps or public domain maps like NRCS. We can use different tools like foliar and soil analysis to guide some of our recommendations. We can use visual symptoms, and in some cases there may be some empirical and process models that might help us appreciate what the stand nutrient demand is at a point in time, and also what the soil supply rates are. Fundamentally, we certainly need to corroborate a lot of our interpretations with field trials, and we do have a wide array of field experiments throughout the South to help really develop these kinds of fertilizer prescriptions. And I always like to tell folks that if all else fails, there are other ways that you can kind of think about getting some assistance. So Sister Rachel's pretty popular. Coming back to fertilizer responses, in some cases simply knowing the soil series may be enough to at least put you in the ballpark, especially soils that have never, in this case, received any kind of phosphorus fertilization. So it's plotting volume. In this case, it's cubic meters per hectare. If you multiply that by about 14.29, you can get cubic feet per acre. And phosphorus in terms of kilograms per hectare, and those are roughly equivalent to pounds per acre. And this is certainly applying phosphorus in higher rates than we would commonly use operationally, but it was an experiment to try to appreciate where the operational limits might be. And in this early work on the Bladen soils, for instance, these are CRIFF A group soils, we'd be putting out about 50 pounds of elemental phosphorus at time of planting to elicit a good growth response, and a little bit less growth potential with the Rutledge and Pomelo soil series as an example. But in addition to knowing the soil series or the CRIFF group, we can use other tools to help guide us in terms of understanding our soils and nutrient supply, and foliar sampling is one. Not too difficult to do, depending upon the size of the trees. We typically will collect foliage from the upper third of the crown, typically fully elongated needles. If you're going to do any foliar collections for micronutrients in particular, you want to make sure that you wear latex gloves, because if you've handled your car keys before you strip off those needles, likely you've contaminated with about all the copper and zinc and other things that that would certainly mask any interpretation. So foliar sampling is another tool that we use, and really the premise of relationship between growth and foliar concentration is this. If we have increasing growth on the y-axis and foliar concentration of a particular nutrient on the x-axis, there is an obvious or pretty significant zone of deficiency, and typically the trees are going to reflect that not only in terms of the foliar concentration, but also in terms of their growth potential and the color of that foliage. There is a critical zone where, again, you're kind of right in that zone of sufficiency and deficiency. Many of these types of soils and stands could in fact respond to fertilizer, but really foliar analyses aren't known with perfect exactness. It's not a real quantitative metric. It's more qualitative. It tells you when something is obviously sufficient or perhaps obviously deficient. So it kind of corroborates your visual symptoms and what you know about your soils. There is a kind of a narrow area of optimum supply or foliage concentration, and then there's something we call luxury consumption, where the plants are taking up more than they actually require. And potassium is one that is a pretty good example of an element that's taken up in quantities that are in excess of what the plant demands. Of course, if we apply too much of anything, we can encourage toxicity, which isn't desirable. The next two slides kind of show you some foliar nutrient guidelines, and these are just guidelines. Again, recognize that they're not known with perfect exactness, but you can see for nitrogen, about 1% is the critical level for slash pine. It's about 1.2 for loblolly. And if you look across these two columns, you'll see that the critical concentrations are higher for loblolly than they are for slash. And that principally relates to the fact that loblolly tends to be a more nutrient-demanding species than slash pine, and we often think of it as one that has higher responsiveness to fertilizers than slash pine, a little bit more plastic species in terms of its ability to acquire and utilize those nutrients. I've also put this slide in for you just to give you some appreciation of what the critical levels might be for some of the important micronutrients: boron, manganese, copper, zinc, and iron, and some examples of deficiencies, which I'll show you in the next couple of slides. So if we go into our site and we know a little bit about our soils and we look at the growth potential of those trees and we're wondering if nutrients are limiting, yeah, we can use visual symptoms as well. And generally, trees reflect soil nutrient supply pretty well, at least in our part of the world. And these visual symptoms are often expressed as general chlorosis of the foliage, in the case of nitrogen, overall yellowing of the foliage. Phosphorus, you might see short tufted needles on the ends of branches. If it's a boron micronutrient deficiency, we might see dead buds at the ends of the branches, a little bit of rosetting. Potassium is very bright lemon yellow kinds of needle colors, especially in the spring on older needles. Magnesium, we don't have a big problem here, but in other parts of the world you might see yellow tipping on trees that are experiencing magnesium deficiency. And copper is kind of interesting because the trees kind of lose their apical meristem and get quite twisted in terms of the branches and form. So if we look at an example of phosphorus deficiency, this is just a picture I took with slash pine. You can see this tufted nature of the crown that I talked about. Very sparse crown. This is a 25-year-old slash pine, probably about 4 inches in diameter. So not only does the sparseness of the crown, but also the diameter and growth and height of that tree depict that something's wrong. But in this case, if you did foliar analysis, we'd probably also see that we're probably well below 0.08 to 0.09% phosphorus in slash pine. Here's that lemon yellow and older needles in loblolly pine exhibiting potassium deficiency. Now these are pretty classic slides that I'm showing you in terms of deficiency expression, and sometimes if we have multiple deficiencies, again it gets a little bit more masked because it gets multiple things are limiting and not just one in a very significant way. Copper. This is one that we've seen in some of our intensively managed stands, especially on wet soils. This is this curly Q aspect of a lateral branch. It's kind of lost any kind of apical dominance, and this is pretty typical of what we might expect copper deficiency to look like. In the stand as a whole, you can see these trees aren't exhibiting very good form. So if we had applied some copper early on, which we did in these stands, we were able to correct a lot of these problems, but you don't want to wait too long. If you have a boron problem, this is some of the rosetting of the buds that you would see. And typically boron is going to be more expressed, especially during droughty periods of the growing season. So if you start to see some of this in an extended droughty period, it might be an indication that you might have a boron problem. And again, you could confirm it with foliar analysis as an example. Ultimately, we use field trials to really help us appreciate what nutrients are limiting, how much of that nutrient we need to apply, and ultimately how much growth response we might expect from those kinds of nutrient additions. And I like to show a slide like this because you don't need statistics to see what's going on. It's ocularly significant. So you obviously have very big, tall green trees here and very short green trees here, and really the difference is the nutrient history in this stand. So some of our fertilizer responses are very dramatic. And it's really in the 1970s, really 60s, late 60s and 70s that scientists in the U.S. South discovered the importance of phosphorus deficiency. And it was kind of serendipitous in terms of how that happened. But this is a shot on a CRIFF A group soils. These are highly deficient phosphorus soils if they've never been fertilized, especially if they've been in forests historically. And it's showing you in this particular example of slash pine how that tree grew for the first 16 years without any phosphorus. Phosphorus was added at a fairly low amount of about 40 to 50 pounds per acre of elemental P, and it grew virtually that same amount of diameter increment in the next 3.5 years. This slide might be a little bit difficult to see, but it's showing you phosphorus, the control plots here in a phosphorus experiment. The trees right in this portion of the slide have not been fertilized. These in the background have received, again, that 40 to 50 pounds of elemental P at establishment. So very dramatic kinds of responses. More examples of the same on some of our B group soils that are phosphorus deficient. And it isn't just the poorly drained soils, as Ed talked about yesterday. Some of our upland soils can also be phosphorus deficient. But as little as 50 pounds of elemental P at age 25 increased the volume from about 2,043 to in excess of 4,500 cubic feet in that same stand. So this is just adjacent plots. So pretty dramatic changes. Another example on an A group soil, no fertilizer weed control on a 13-year-old loblolly in Gulf County, Florida. This is a representative dominant tree in the control plot, and this is a representative dominant tree in the plot that received fertilizer and weed control. This is pretty dramatic. We've gone from 433 cubic feet per acre to an excess of 4,200 cubic feet at the same age. So a tenfold increase. That doesn't happen all the time, but it certainly happened at this site. So if we had to think about typical application rates for fertilizers used with our southern pines, and this might be variable again depending upon the specific soil that you're on or your physiographic region or history of land use. If you're talking about time of planting fertilization, most commonly we're putting out some combination of N and P, 35 to 50 pounds of elemental N, 25 to 50 pounds of P, and oftentimes applied as diammonium phosphate or monoammonium phosphate. If potassium is limited, and there are some geologic terraces where we might expect some potassium deficiencies, you might be putting in some levels of potassium following these general rates. Again, I wouldn't take these as the only rates that you might apply. It might require less or it might require more depending upon your site conditions. If boron is limiting, we never apply more than a pound per acre, typically in excess of a pound per acre of element, which isn't much. can lead to toxicity. So you have to be very, very careful with especially the micronutrients. If you're kind of phasing out of the time of planting options for fertilization, you're working with more mid-rotation closed canopy stands, we find that really our best responses tend to be a combination of nitrogen and phosphorus, anywhere from 175 pounds of elemental N to 25 to 50 pounds of elemental P would be kind of our general prescription rates. So we use different common fertilizer sources, diammonium phosphate, monoammonium phosphate, be common N and P at time of planting treatments. If we use less triple super phosphate these days, but it has been used historically. We've also used ground rock phosphate historically. I'd say in terms of our nitrogen sources, urea would be the most common, and if you're working with any type of potassium, probably potassium chloride would be the most common. So an important thing to recognize here is the guaranteed analysis. And I can't tell you how many times people make mistakes in terms of interpreting that guaranteed analysis. The first number is N, the second number is P, and the third number is K. But the important thing to recognize is nitrogen is the only one that will tell you what the percent nitrogen in that material is. So 18 says that diammonium phosphate contains about 18% 18% nitrogen. The 46 represents the percent P2O5, the oxide form of phosphorus, not the percent phosphorus. So in order to know how much elemental phosphorus is in that fertilizer, you have to convert from oxide to elemental by multiplying this 46 times 0.436. Likewise with potassium, the third number, that's always expressed in a guaranteed analysis in the oxide form. So you would have to again convert that by multiplying the KTO value by 0.883 to put it in a percent K. Very important thing when you start determining how much fertilizer you put out. In fact, most of our fertilizer prescriptions really often talk about elemental rates because you might use different fertilizer sources to reach that fertilizer prescription. So be aware of those types of things. So when you're doing a fertilizer calculation, we're just doing an example with diammonium phosphate here. If it's 1846.0, if you put on 250 pounds of diammonium phosphate, that would be equivalent to 45 pounds of N and 50 pounds of P. So to do the calculation, 250 times 0.18, which is the guaranteed analysis of nitrogen, equals 45 N. If you're working with phosphorus, it's 250 times 0.46, which is P2O5, times 0.44 to put it into an elemental phosphorus form. So always keep in mind that phosphorus and potassium are listed in the oxide forms. Urea is fairly easy. It's 4500. You can read nitrogen directly. So 335 pounds urea is equivalent to about 150 pounds of nitrogen. So here's some growth responses that we might expect in mid-rotation fertilization. These are data, historical data from the former North Carolina Forest Nutrition Cooperative, now called the Forest Productivity Cooperative, and the CRIFF program at UF. And what this graph is showing is volume response over 8 years to differing treatments. So this upper red line is Loblolly response to varying levels of nitrogen with 50 pounds of phosphorus. And slash pine is the same. This is varying levels of slash pine response to varying levels of nitrogen with phosphorus. The bottom graph, or bottom line, is showing you responses simply to nitrogen alone without any phosphorus. The take-home message here, and this is pretty consistent across a wide array of sites, is we always get a better response to N plus P than either nitrogen or phosphorus alone. And that's to maintain balance and in fact ensure that we're meeting the demand requirements of the species. So if we look at this graph, clearly you can get better responses if you up the ante in terms of the amount of nitrogen, but affordably we don't do that, and that's why our upper limits of nitrogen are often restricted to a maximum of about 200, because that makes it economically feasible. So the reason why fertilization works in terms of providing good responses in stands is depicted in this graph. So this is plotting simply trees per acre by diameter class for different fertilization regimes. And this is what the stand looked like at the time of fertilization. So the typical bell-shaped diameter distribution for a even-age slash pine stand. This graph shows you what the control plot did some years later, 5 years later. And naturally, we'd expect that diameter distribution to shift to the right. The fertilized stand that received the N+P shifted that diameter distribution even further to the right into producing larger size trees. And when you can shift product classes from pulpwood into chip and saw into saw timber, of course the value of that stand rises considerably. And that's what makes a lot of these types of treatments affordable. So in some cases the fertilizer responses are quite dramatic, as we've seen in other slides. This is a typical flatwood site. This is a control plot in a Gallberry flat. It's a C group soil, CRIFF C group, so Spodosol with clay. 4-year-old loblolly pine growing on the control. The same age stand growing with weed control and fertilizer in the background. So pretty dramatic. This is the same, essentially the same picture taken at age 19. Pretty remarkable. This is the control tree here, and this is the fertilized tree that received a combination of fertilizer and weed control. So to come back to the first question, can we change site index, kind of the potential productivity of a site? The answer is absolutely. And these are actual measured data, not estimated estimates of site index, for an experiment that we had near Gainesville. And the control plot at age 25, in this case for loblolly pine, averaged about 64 site index. Where we used weed control alone, no additional fertilizer, we increased site index by about 10 feet. When we used fertilizer only, without any additional weed control, we increased the site index by about 20 feet. And the combination of fertilizer and weed control, the site index is about 87. So when you kind of take your site index data and convert it into yield tables, you can appreciate that a 20-foot difference in site index is significant in terms of the amount of growth. And this slide is one that appeared in the 2010 Journal of Forestry article. Citation is listed below if you have interest. And the take-home message is this: nutrient management, again, either by fertilization or weed control or combination, certainly can increase stand value. And it does it largely by increasing not only the amount of volume, but also the amount of grade material that's produced in that stand. So let's look at loblolly pine in this case. This is plotting stand volume against diameter. And it's showing you the total amount of volume produced in that stand at age 25, about 2,370 cubic feet. Of that total volume, we merchandised it by size class, and about 61% of it is in pulpwood, and about 39% of it might fall into what we call grade, chip and saw or saw timber. If we look at the maximum treatment, where we added fertilizer and weed control, and the fertilizer was added more than once during this experiment, you can see that we've more than doubled, or we've about doubled the amount of volume produced, but we've clearly changed the distribution of that volume in that stand to higher value material. So in this case, pulpwood only constituted about 14% of the volume, and chip and saw timber was about 86%. I might add that this is also these are unthinned stands, so had we thinned it at an appropriate time, some of these differences might have been even more pronounced. But the message is that certainly you can change productivity and value of that stand if you understand the limiting factors for that site. I mentioned micronutrients. Subacute stresses can be induced by intensive management. The supply in the soil just isn't adequate. And if we're causing these stands to grow rapidly with weed control and NNP, clearly you can kind of induce a deficiency of some of these micronutrients, and typical to what I showed you with copper previously. But in one of our earlier experiments with manganese, it received nitrogen, phosphorus, and potassium. By simply adding manganese, relatively low amounts, we increased volume growth by another 32 cubic feet per acre per year above the control. So the nice thing about most of our micronutrients is that they persist for long periods of time. And if you recognize that you've got a micronutrient deficiency, if you take care of it fairly early, either by applying a specific micronutrient as part of your blend or more of a prophylactic treatment of like a 10-10-10 plus micronutrients, you may in fact reduce these types of deficiencies from occurring. So I put this slide in simply because it's a story a little about copper, and we published a paper here recently that looked at loblolly and slash pine responses to a variety of micronutrient additions. And what we did was load up the treatments with some nitrogen, trying to induce some deficiencies, as well as phosphorus and potassium. And then we used the minus element approach to try to identify which micronutrients were limiting. And suffice it to say, what the results suggest is that the Loblolly pine site clearly responded to copper, whereas the slash pine site did not. And what you see in this graph is the malic extractable phosphorus concentrations in the soil by horizon, so going from the surface all the way down to the argillic horizon for both of these sites. And the interesting thing to note, if you look at copper in this particular case, which was limiting, it's very low supply at this location across all of the horizon depths. In contrast, at the slash pine site, the copper extractable, malic extractable copper supplies were relatively low on the surface horizons, but sizable amounts were present in the lower subsoil, so the BH horizon and the BT horizon. So this site did not respond, and this one did. And this is just one example. Of course, we'd certainly like to test this across a wider range of sites, but it certainly indicates that supply, even though these are very similar, taxonomically similar soils, they're both Spodosols with clay, a C group soil, they had quite different soil nutrient supplies. So a word of caution that just because the soil looks the same doesn't mean they are the same for any of the nutrients that we're talking about. You can overdo it in terms of nutrient additions, and I put this slide in there to let you appreciate that. You can either do it from nutrient additions that you've prescribed in terms of NNP or other macro and micronutrients, or you can have it sometimes happen inadvertently from other types of operations. This is a picture of a poultry operation, and these are tunnel ventilated systems, so they're blowing out a lot of particulate matter and ammonia in particular out of the fans at both ends of these houses. And you can see that there's a lot of dead and dying trees, particularly in the cone of influence where those fans are blowing out. Pitch canker is certainly a disease that is responsive to nutrient imbalances and excess nutrients. And you can see that a lot of that mortality occurs in this portion of the stand. It even occurs back here, but less so as you move a little further away from that location. So the message here is that this is not the kind of stand that we'd like to produce. And in this particular case with slash pine, you can do that with excess fertilizer additions, but it also shows you in this case what nitrogen levels were relative to proximity to these poultry house locations. So the darker colors would be higher nitrogen concentrations, and as you move away from those sources, of course, N levels go down, and so do the levels of disease and mortality. So a lesson to recognize the importance that more isn't always better, and you have to appreciate that some species, and in fact some genotypes, might be much more susceptible to these types of concerns. We use soils, obviously, to also help us with our site preparation decisions, and of course it's pretty obvious to folks that have worked in poorly drained soils that bedding is a very integral part of our management system, not only for creating an elevated location for the seedlings to be planted, but also causing some mixing of that organic matter that was left after harvest into the soil that might foster increased mineralization and nutrient supply. And a simple mechanical treatment like bedding, in this case, might have a pretty profound effect in terms of growth as well as survival. We've probably all seen a lot of these kinds of stands where you've got standing water and trees aren't too happy unless they're a little bit above that. We've also used a lot of mechanical site preparation historically to kind of simulate old field effects, and these are some site preparation response data from the Piedmont, and you can see that if you increase the intensity of chopping and in some cases shearing or harrowing, you can increase productivity. Now, a lot of these sites, we've probably gotten away from a lot of this heavy mechanical. We're using more chemical site preparation to accomplish a lot of the same goals. This is actually a ripping plow if you have some kind of an impervious horizon that has been used on some locations to increase the amount of rooting depth for those trees. Of course, the impacts of competing vegetation is well known these days, and some of the early work done by the Auburn University Herbicide Cooperative kind of paved the way for a lot of other technological advances to occur. But in this particular case, you can see what herbaceous weed control does. You got a sea of andropogon here, and where you've done a directed spray over the bed, the trees are pretty obvious. They're less so when you don't use that kind of spray. And those treatment responses can be pretty long-lasting, as I showed you in an earlier slide. Simply controlling the weeds alleviates nutrient stress almost to the same degree as fertilizer additions in some sites. This is a bit of data showing you on a CRIFF A group soil, these wet gumbo clay soils with clay near the surface, some differences in response between slash and loblolly pine. Control is C, F is fertilizer, 250 pounds of diammonium phosphate. H is just an herbaceous weed control treatment, and FH is a combination of fertilizer and weed control. And you can see kind of files that generality I described earlier. Loblolly pine tends to be more responsive than slash pine to any of these types of treatments. And on these kinds of A group soils, loblolly pine, if your objective is to grow trees for profit, is often the species of choice. And it does very, very well on those types of locations. These are just a couple of additional slides to show you what stands might look like through time. This is a 23-year-old slash pine stand. Received a single bed pass on a poorly drained flatwood soil, probably a C group soil, and one mid-rotation fertilization, 2,100 and change cubic feet per acre at age 23. A site that received double bedding, pretty significant competition control, and 3 fertilizations had in excess of 3,400 cubic feet at age 13. So one of the last things that I want to talk about, or one of the last, close to the last things I want to talk about, is genotype by environment interactions. And I bring this up because we're deploying different genetics these days. We've gone from open pollinated types of deployment to full sib kinds of deployment seedlings, and in some cases even varietal seedlings. And genotype by environment, this term, the genotype by environment interaction, is really describing variation in the relative performance of different genotypes from one environment to the other. So if we look at this graph, it's showing you increasing crop yield on the y-axis and increasing site resources availability on the x-axis. So we might think about this as increasing site quality and relationship to increasing growth. So if we look at genotype B and C in general for both, we're seeing that you get increased growth as resource availability increases for both species. Genotype B is more productive than genotype C. So that's interesting to note. Now, not all genotypes behave in the same way. In this particular graph, it's showing the same thing, increasing crop yield relative to increasing site resources. And it's showing you that genotype A is very responsive, much more so than genotype C, to increasing site quality or increasing resource availability. It's this type of non-parallel lines that really would characterize a genotype by environment interaction. It's not only a biological effect in this particular example, this hypothetical example, but it's also a statistical effect that if you analyze the data, you would be able to detect that interaction. If we look at this particular slide, it's showing you relative to genotype C that increases with increasing site resources. Genotype D did the opposite. It did not do well relative to increasing nutrient supply. And you might ask, well, what's causing that? It could be that you've increased the susceptibility of that genotype to various damaging agents, like fusiform rust or pitch canker, like we just described, or it could be any other number of adaptation characteristics of that particular family. So I put this out there to let you appreciate that don't assume if you buy loblolly pine seedlings that they're all the same, or slash pine seedlings, or any species for that matter, that different genetic sources might behave differently on different sites, and also in response to different types of treatments. So I show you a little bit of data from one of our experiments, age 12 data from two contrasting soil types. One is a CRIFF A group soil, poorly drained gumbo clay soil, and the other is a CRIFF D group soil, so it's a spodosol with clay. And it's plotting stem volume for low culture and high culture. Low culture has received a little bit of weed control at time of planting, some early DAP treatment and mid-rotation treatment. The high culture received pretty aggressive weed control and also multiple fertilizations. And you can see that some families are not behaving the same. In this particular case, L5 just didn't do the same thing that families like L4 did when you increase nutrient supply. So again, same type of site, but different responses of the different genetic sources. This is another site, and you can see that this same family, L4, was the top performer on this site. Quite different soil type, but it did very well when we increased nutrient supply through, in this case, weed control and fertilization. Some other families are not as responsive. So again, this is where we're at relative to some of the current research. We don't have generalities for all families or locations yet, but this is something that I just bring to your attention as an issue that you should keep in mind. The last thing that I'll mention and finish up here is that it's great to be thinking about the fact that we can increase the amount of growth associated with different silvicultural treatments and investments, but we also have to be thinking a little bit about what's happening to that site relative to long-term carbon storage, not only in the above ground portion, but also the below ground components. So here's a site that was about age 20. It's showing a control plot with quite a mixture of understory present, fairly skinny trees for that age, and another plot at the same location where we attempted to control the weeds fairly aggressively and also add fertilizer. So clearly big differences in terms of the above ground component and also what's present in the understory. At age 25, we kind of did a carbon and nitrogen balance for this location. I'm only showing you carbon accumulation at this in this particular slide, but it's looking at forest carbon pools, both in the soils. This lower portion of the slide is the soils. The litter or detritus is this grayer portion, and the little darker gray would be the vegetation responses. And this is for both slash and loblolly pine on the same site. And you can see that the axes are exactly the same in terms of scale. The treatments are control, fertilizer only, weed control only, and the combination of fertilizer and weed control. And similar to what we described earlier, we're seeing a pretty good response to, in terms of total ecosystem carbon gain to the fertilizer treatment. A little less so, of course, with the weed control only, and a fairly sizable, but probably not a significant difference when you combine the fertilizer and weed control. Pretty similar story for slash pine. So the vegetation patterns that we're seeing here generally followed some of the basal area data that we also observed, but if we look at total ecosystem carbon, the greatest amount is with the fertilizer only. And you can see is that fertilizer is not only increasing the total in the ecosystem, but also seems to elevate the pools in the soil. And that's true for both loblolly and slash pine. We see a little bit of reduction in those plots that received aggressive weed control. And one might surmise that you have less roots that are probably turning over in those kinds of systems. And really the long-term kinds of data, kind of looking at the effects of these types of treatments relative to ecosystem carbon accumulation are relatively few. So I bring this up only because it's an interesting scientific question for us to consider, and we have actually remonumented this particular experiment. We've maintained the same plots, and now we've replanted it with trees to look at what the carryover effects of some of these fertilizer or weed control treatments might be, not only in terms of tree growth, but soil carbon sequestration, as well as what's happening in terms of reinvasion by understory plants, either in the actively managed or the untreated carryover. So I raise this point again just to let you appreciate that we need to be thinking about the long-term impacts. We all certainly have the objective of having all of our forestry treatments be sustainable, and clearly these types of studies are important to help demonstrate that. And I can tell you right after at age 3 with the remonumented plots that virtually all of these treatments are doing better in the second rotation than in the previous rotation, even the control plot. So we've changed the genetics to a certain degree. We've elevated the amount of carbon dioxide in the atmosphere by about 50 parts per million. In our embedding operations, we've incorporated some of the organic matter and debris and so forth. So a lot of that might be contributing to some of the growth responses that we're seeing. But the positive point is on this one study that the growth of the next rotation is actually better than the previous. So with that, we've covered a fair amount. I'd be certainly happy to open up the discussion for any questions that you might have, and I'll do my best to try to answer them for you.
While we're letting everybody talk or type their questions out, we did get a few earlier on in the session. I'm not sure he's still present, but Jake Meyer asked, I thought soil index is fixed per species and soil type. Fertilization and other measures change the soil type, I thought.
No, they don't really change the soil type, but if we think about kind of the inherent site index, that's perhaps what you're thinking about relative to that concept. But it's often as useful to think about what the potential productivity of that site might be, giving the silvicultural treatments that we might impose. So, yeah, it's probably best to think about site index or site potential is not being fixed, at least in our part of the world where we recognize that there are truly some limiting factors that we can control silviculturally in terms of our management systems.
We had a second question from Toby. Is using fertilizer a timber expense or a land expense for IRS capital gains?
Good question, Toby. I'm not an economist, so I'm a biologist, so I'm probably not the best to ask for that, but I think that probably most organizations will work that out specifically with IRS, whether it's capitalized or expensed. So I direct you to an economist on that. I'm sorry. The other thing that I'll point out, if there's any other questions, is that Sean has posted a URL for an extension publication that I put together with Alan Long here at the University of Florida. It's called Using Soils to Guide Fertilizer Recommendations for Southern Pines. So it goes through a lot of the types of things that we've talked about today. It's actually taken the CRIFF soil groups, and at least for Alabama, Georgia, and Florida, I've converted all of the main soil series that we have in these states to CRIFF soil groups for you. So there's a lookup table that might prove useful to you as you peruse the literature and try to make recommendations for your own land types.
And we have another question from Toby. Would uprooted trees from a microburst and a salvage timber harvest using a TIMCO cultivate the soil and cause a release of nutrients?
Good question, Toby. It depends on the level of disturbance. I mean, there's nutrients contained in the subsoil, and you may see some microsite changes in terms of nutrient availability, but generally it's going to be little more significant kinds of site preparation activities like a disc harrow or bedding that's probably going to cause greater incorporation of the surface organic matter and facilitate mineralization decomposition of that material and converting it from organic to inorganic form. So the answer is yes, but probably it'd have to be done in a little bit more directed fashion to have much impact on tree growth except at a local level.
Well, if there aren't any questions, and it doesn't appear that there look like any other people typing, for the sake of time, we are going to wrap things up. So thank you again for everybody for coming in and joining us. This was made possible by the Forestry and Natural Resource Webinars, a partnership between the NC Cooperative Extension, SREF, and the Texas A&M Forest Service. And just a reminder, if you were interested in receiving CEUs, you should have got a pop-up window, but if not, a link was posted just above in the chat box that you can follow through to take a quiz on today's content and receive your credit. And if everyone would give Eric a round of applause, I think that'd be great.